Bloodstream infections are a really serious condition, and when you present with symptoms of a bloodstream infection, every hour that elapses that you don't get the right therapy increases your chances of dying. The technology that we're currently using to diagnose bloodstream infections is old. It relies on the growth of microbes on agar plates, and ultimately it can take anywhere from 2 to 5 days in order to get lab results back.
So we capitalize on the fact that microbes secrete and consume metabolites much faster than they multiply by developing a metabolic preference assay, which measures the rates of these consumption and production markers in order to identify microbes, as well as measure their antibiotic susceptibility profiles. So the nice thing about our strategy is that we can perform an assay in a matter of hours, usually about 4 hours, versus days.
The BSI-Dx is different from other kinds of platforms because it was designed for large-scale diagnostic labs. A single platform can support a metro area of more than 2.5 million people, and it's flexible to essentially any kind of microbe that can be grown in culture. This platform is borrowing from more than a decade of development in clinical chemistry and allows us to detect these microbes in a fraction of the time.
It allows the technician to go from sample straight to result with a push of a button. The MCD is a custom plate that we developed in-house to separate the sample from the analytical chamber to allow us to real-time sample off the mass spec. By using the MCD, we are able to bypass the sample preparation that is required for processing blood bottles. So we can take samples straight from a blood bottle, put it into our system, and get results directly.
One of the main challenges that we face when doing metabolism studies is that various media can have a lot-to-lot variability. This can actually influence our metabolite patterns. We have worked with our team to develop a custom medium which allows us to stabilize metabolic phenotypes from run to run. So we tested the efficacy of this media in large-scale clinical trials and found that it is robust, reproducible, and a cost-effective strategy for identifying microbes and testing antibiotic susceptibility patterns.
My role in developing BSI-Dx has been to develop a platform that's able to deal with the needs of a population of over 1 million people on one platform. We selected Thermo Fisher's Altis MD, which is an off-the-shelf Class 1 medical device, which is useful for us as it helps us accelerate our regulatory pathway. It's an extremely powerful triple quadrupole mass spectrometer that's able to detect 1,000 compounds per second.
We are currently validating our technology with our strategic partner, DynaLife, in order to get the data that will allow us to submit for Health Canada and FDA approval.
If we put this BSI-Dx platform into action here in North America and Europe, we'd be saving as many as 50,000 lives a year from people who are currently dying from slow diagnostic timelines.